So our next speaker, another very experienced researcher in the bat world. We've got Dr. Leigh Richards, who will be talking about basically specimens, which we have mentioned previously. She's from Durban Natural Science Museum, and her talk is entitled Natural Science Collections-Based Research, the Good, Not So Good, and Downright Amusing. If you can go ahead with that, please, Leigh. Thank you.
Good day, everyone. Thank you for joining my talk. I am Leigh Richards, Curator of Mammals at the Durban Natural Science Museum, Africa, and lies at the center of the Mpumalanga-Pondoland Albany Biodiversity Hotspot. The museum houses a modest mammal collection, mostly comprising small mammals from Southern Africa. It hosts a collection of around 4,000 bat specimens belonging to 126 or so taxa, with notable specimen contributions by the former curator, Professor Peter Taylor, and long-time benefactor, Professor Adam Monaghan. These 126 taxa are roughly a third of the known or previously described African bat biodiversity, which was estimated just shy of 350 species as at 2020-2021. Most African bat species were described between the 18th and early 20th century. However, in more recent times, we have seen a dramatic increase in the rate at which new species are being described or taxa resurrected from synonymy. And there's still more to come. From the late 80s until more recently, at least 60 species have been described, if not more, from the Afro-Malagasy region. DNA analyses have been a major contributing factor to these discoveries and has become an important first step in unraveling the often complex evolutionary relationships among our beloved winged creatures of the night skies. In the context of South African bats, Taylor 2000 reported 52 species. However, with the help of systematic studies employing DNA sequence data analyses, several widespread species have since been split, leading to an increase of 61 recognized South African species as detailed in Monaghan et al. 2010. Currently, the estimate sits at 66 species for the country. However, accurate evolutionary reconstruction and species description relies on multiple lines of evidence. These are a few recent examples of species description papers that have employed such an integrative taxonomic approach. An important component of such integrative taxonomic studies are scientific specimen collections. Specimen collections and associated derivatives are an invaluable resource in taxonomic and systematic studies, having enabled the refinement of species boundaries and a better understanding of the historical diversification. However, as time has marched on, there has been an increased call to consider the potential impacts of collecting voucher specimens on wild bat populations, with some proposing alternate non-invasive sampling techniques and the vouchering of specimen derivatives in lieu of whole physical specimens, especially in the case of rare species or those of special conservation concern. Others have highlighted the importance of such collections in their continued support, as they can contribute to important studies, such as infectious disease investigation. Being mindful of the sensitivities around the collection of voucher specimens, the Durban Museum has looked to additional means of augmenting the growth of the collection. The museum now serves as the South African Bat Assessment Association, also known as SABA, recommended biorepository for bat carcasses received from operational wind energy facilities in the country. Procedures concerning the treatment of bat carcasses are contained in the South African Good Practice Guidelines for operational monitoring for bats at wind energy facilities and is available at the SABA website. Through all the effort to deposit bat carcasses with the museum. Well, Ecological or environmental consultants are provided with reliable and/or verifiable taxonomic identification of the donated bat specimens. The donated specimen material can support other research studies. The biobank tissues support various genetic-based studies. And museum staff can conduct necropsies and provide information on plausible cause of death. Museum staff can also gather additional biological data that can hopefully feed into informing mitigation efforts. And lastly, the carcasses and the derivatives are safely processed and or disposed of at the museum facility. With respect to the first benefit mentioned, accurate taxonomic identification is not always possible by consultants using external morphology alone. Specimens typically come in with varying degrees of decay or deterioration. Specimens such as these are almost impossible to identify without expert examination of cranial and dental morphology. The museum, in collaboration with Sabah, Nkululeko Wildlife Service, and Richardson and Peplow Environmental, initiated a pilot study examining trends amongst almost 200 bat fatalities donated to the museum between 2015 to 2018. These bat carcasses emanated from 7 of the then 26 operational wind energy facilities. We found 7 species belonging to 4 families. Tadarida aegyptiaca, the Egyptian free-tail bat, and Liphotus capensis, the Cape Seratine bat, accounted for 91% of all examined specimens. One may argue that these species are of least concern, both in a regional and global conservation context. However, an unpublished study by Reddy showed that Southern African Egyptian free-tail bats in fact comprise 2 distinct morphospecies as based on cranial morphometry. There's a larger, more robust taxon, most likely, or morphospecies, most likely Tadarida aegyptiaca, and a diminutive western morphospecies that possibly meets with the description of the subspecies Tadarida aegyptiaca bourgeois. DNA studies are presently underway to determine whether these represent separate taxa. And based on material from biobank material from specimens derived from wind energy facilities, further genetic studies are being done to determine whether which of the 2, or perhaps both, if there is such, are being afflicted most by the wind turbines in South Africa. Hopefully, we'll have further details to share in upcoming months. Going back to the original study of the sample sizes, we decided to assess the relative age and sex of the specimens. Where possible, the reproductive status of males and females were recorded. Relative age was determined from the degree of tooth wear of the maxillary canine, the first premolar and the first molar of Egyptian free-tail bats and Cape Seratines. 4 age or tooth wear classes were recognized. To our surprise, and based on available specimen material, species-specific trends were indeed noted. In the case of Tadarida aegyptiaca, males formed the majority of the samples examined from 5 different wind energy facilities. In the case of Liphotus capensis, older individuals with greater tooth wear were more often noted in the sample than individuals bearing a lesser degree of tooth wear. This study is ongoing, and I have a few more 100 samples to get through. It will be interesting to determine whether the previously observed trends from the pilot study persist. We do encourage consultants to donate bat carcasses. However, we have noted that there have been some challenges in this respect. Onerous regulations, necessary authorizations, and permit requirements make it challenging for researchers, but perhaps even more so for non-researcher donors, such as environmental consultants wishing to donate bat carcasses. This is part and parcel of my work life as the curator of mammals. And if you walk into my office, you'll be greeted with a bookcase full of compliance documentation relating to collections. Challenges specific to the donation of bat carcasses from wind energy facilities include long permit processing times, short validity periods, and a paucity of valid contact information. Sometimes consultants do not even know where to obtain the necessary documentation or where to submit completed documentation. The specimen material itself must be treated, packaged, and couriered according to applicable regulations governing the movement of animal material and road and air freight. And some permits issued to consultants limit the effectively they're hampering their donation to the museum for use by other researchers. When such challenges start to weigh me down, and I feel like I'm taking a knock from having to deal with all the red tape, I turn my thoughts to all the perks associated with a career such as mine. Visiting breathtaking sites and trekking through forests and savanna woodlands in search of bats certainly can refresh one's soul and make you ready to tackle all the red tape. And of course, with all of this comes some unusual and funny experiences, such as getting stuck in the mud at field sites, having to dig your vehicle out and so forth, or lost in the forest for an hour or two as it's approaching midnight, or having our bat sampling session in a reserve set up across a pool, gate crashed by a herd of ellies. Footage taken from 2011 at a private game reserve at which we were conducting a bat survey. Apologies for the grainy image. Note the bat equipment hastily disassembled and strewn on the deck by the pool. And then getting to interact with an amazing lot of postgraduate students who teach me something new each time that we are in the field together. I really look forward to seeing where this new generation of bat scientists end up and make their mark in our field of study. And in collaboration with organizations such as the Bat Interest Group of KwaZulu-Natal, conducting training relating to bats for conservation officials and also those in the pest control industry. Following on from my postgraduate student supervision, this is another really fulfilling and rewarding interaction that I get to experience as part of being a curator at the Durban Museum. However, none of this would be possible without the contributions of a few Wonder Women associated with the museum's mammal department. From the technical assistants who over the years have overseen specimen preparation and overall curatorial care of the collection. The current staff member being Miss Summer Welasi, featured at the top left, who, despite having to work through bags full of wind farm bats, never complains. A heartfelt thanks for your dedication to our department. The interns who have come and gone, and the ad hoc assistant, Mrs. Kate Richardson, who has provided invaluable assistance over the years. Amazing team of Natural Science Museum volunteers, without whom we would not have and all that we do in terms of our collections. We thank you. And thank you, esteemed listeners, for your attention. My best wishes to our future women in the Natural Science Collections facilities throughout Africa. Ngiyabonga.
Thank you very much, Leigh. That's great. We do have a couple of questions for you here. And as our last but not least speaker, the first question actually is very relevant. We've got Anindita who's asking, thank you for the wonderful presentation. Can you please share your experience as a woman in the field of Chiroptera study? You're on mute. Sorry, Leigh.
Good day, everyone. Apologies. I'm not quite sure what has happened to my lens. I'm appearing all blurry at present. It's not your eyes. It's something to do with my camera. My experience as a woman in science. I think the one thing that I have been very pleasantly surprised by is just how many women are recruiting into fields such as natural science collections facilities. I think we all know that before it's always been a very male-centric type of environment, but certainly in the Durban Natural Science Museum, you'll find that we are mostly women, and very proud of it as well. You know, I think of when we jump into the 4x4 and have to head out to field sites and really put that vehicle to the test. We are holding our own with, you know, with our male colleagues as well. Of course, with that though, as well comes some concerns, and especially when working at night in sites, safety, of course, is a great concern, and I think more so being a woman. And especially when you're a woman, and I'm a mother of two kids. I've got a 6-year-old and a 7-month-old at home. My interns, my assistants, or sort of my kids in the field. I think that there's also added pressure, you know, being a woman, being in the field, feeling so responsible for the young people, sort of under your care, you know. But I think it is an exciting time. There have been some really major advancements in our field, in bat science and so forth, and pioneered by women. So I'm quite keen to see what all you ladies out there are going to be putting forth in terms of the scientific community and expanding our knowledge in future years. Unmute, Helen.
Oh yes. Yeah, so I absolutely agree. There's been some fantastic speakers of all levels of careers here today, and it's just so refreshing to see everybody so excited about their areas of research and also the new things that are happening right now. Maybe we can take one more very quick question from Isabella here. It's a general question about tooth wear, and I know that you're probably a really good person to answer this question. The teeth get worn down quickly regardless of age. We observed that Pteropus that likely eat lots of sugary fruit have pretty bad teeth really quickly.
No, that's the thing. It isn't an exact science, and also depending if you do observe that there are regional differences in diet, certainly that can confound matters. So definitely not a hard and fast rule, but for the available specimen material that we have, it was sort of the best that we could do. But one needs to be mindful, especially as you extend your scope more broadly in terms of geographic scope. Then it lends itself to possible variation in diet, which then could definitely, if there's a population eating harder insect prey, just because that's what's more readily available to that particular population, it will indeed skew the degree of tooth wear relative to others that are exploiting more softer shelled or softer exoskeleton insects.
Thank you very much. Yeah, thank you again for your talk. It was great. And thank you so much to everybody else who has spoken today. And just a very quick thank you to the Bats Without Borders organising team. I jumped in very late, so I absolutely can take no credit at all. So I will, on that note, hand back over to Rachael, who will be concluding the seminar. Thank you very much, Rachael. You're on mute.
No, we've done 3 in a row. We're doing so well. So a big thank you to Helen, who is in the final stages of her PhD, which is why she isn't presenting today, but she did step in for us at the last minute. So I do have quite a lot of thanks, and I hope I've remembered everybody. First of all, a very, very big thank you to Ruth Fraser, who is our admin and comms officer, who has really done the lion's share of organising this event. So huge thank you to Ruth, who's quite new to the team and just started in October. Also to Paul Howden-Leach, who is one of our board members and has been actually doing all the videos and everything in the background. So it's been a very seamless process for us with his techie knowledge. And also, of course, thank you very much to our speakers who have really made this event. As I mentioned, this is the first time we've had a Women in Science event, and this has come out of some quite serious gender inequality issues that have been raised. And so we have a fantastic early careers committee members, of which a number of people on this talk, including Anna and Alex, and Damalera are on the committee. So please do sign up if you haven't already, or go and read up a little bit more about that. As I mentioned, we're going to be doing some networking events, and as has been raised, some of the challenges, but also networking opportunities. And we are going to be also rolling out some early career training program, which is available for men and women as well. And obviously our speakers today have really been fantastic. From Dina is an inspiration to me and many others. We've also been listening about inspiring people. All of our speakers, I think you can admit, are incredibly inspiring and really motivational. Of course, our fantastic bats who have motivated us all. We've also particular thanks to our early career speakers today, who are kind of just finished their masters or doing their PhDs, really inspiring and international standards. We have obviously people represented from Nigeria, Mozambique, Eswatini and South Africa. And then, further on in their careers, we've had Marieke and Leigh. And I actually met Leigh, who had finished her PhD in South Africa as I was just starting mine. So it's really lovely to see people, popping back up in our lives. And Leigh has been not only doing research, but also, as you had seen, is inspiring lots of people and supporting lots of training that we've had through Bat Conservation Africa, but also with Bats Without Borders. So we are really very, very grateful. And of course, to all of you who have come today, it's been a fantastic support. To see so many people online. As I mentioned, we'll be making this available on YouTube, so we will send that out. Because it is our first event, we would really value your feedback and ideas for the future. So we will send out an evaluation form. And if anybody would like a certificate for attendance, there'll be an opportunity for you to sign up for that. And I've also just popped in the chat. So we do run, if you haven't already come across, our Winged Wednesday webinar. So we do a monthly webinar. So this one has replaced our February one, but we do once a month. It's the 4th Wednesday of every month. So that is in the chat. And I think I've hopefully got everybody, but thank you again for a fantastic start. It is International Women and Girl in Science Day tomorrow, so do have a look out. So it's the 11th of February every year. And so a big shout to all the motivating and inspiring women across Africa and beyond. You have done a fantastic job, and we're really proud to hear of all this great work. Thank you very much.